As we saw earlier on in this course, under anaerobic conditions, glucose is broken down into two pyruvate molecules that are then converted to a stable fermentation product. This limited degradation of glucose releases very little of the potential energy of glucose. Under aerobic conditions, the cells can use oxygen and completely oxidize glucose to carbon dioxide in a metabolic pathway called the citric acid cycle. This pathway is often referred to as the Kreb cycle in honor of Sir Hans Krebs, who worked out the steps of this cyclic pathway from his own experimental data and that of other researchers. It is also called the tricarboxylic acid cycle, or TCA cycle, because several of the early intermediates in the pathway have 3 carboxylate groups. Once pyruvate enters the mitochondria, it must be converted to a 2-carbon acetyl group. This acetyl group must be activated to enter the reactions of the citric acid cycle. Activation occurs when the acetyl group is bonded to a thiol group of coenzyme A. A thiol is like an alcohol, but instead of an oxygen, we have a sulfur. So this would be the generic structure of a thiol. Coenzyme A is a large thiol derived from ATP and the vitamin pantothenic acid. It is an acceptor of acetyl groups which are bonded to it through a high-energy thioester bond. The acetyl coenzyme A formed is the activated form of the acetyl group. Here I have my acetyl group. This is highlighted in pink. And when it gets activated in the reaction with coenzyme A, it forms a thioester. And the reason we have this squiggle bond is because acetyl coenzyme A is the activated form of that acetyl group. Again, here's the acetyl group circled in red. You probably remember an ester. So when we have this kind of arrangement of atoms, we have an ester. And when I replace this oxygen, when I replace that with a sulfur, I have a thioester. Thiols, these are functional groups that deal with sulfur atoms. When we take a look at the structure of acetyl coenzyme A, it becomes abundantly clear why we like to use this shorthand form, where we have our thioester and then we write CoA, and this CoA represents all of this. So here's my acetyl with my sulfur atom right here, and the CoA, we represent all of this is represented by just this part here, the CoA. And both of these molecules are acetyl coenzyme A. This one here is just a bond line structure. If we take a look at the one on the top, you can see what acetyl coenzyme A is made from. Of course, you have the acetyl group out here on the far left, but if we go to the far right, you see that we have our phosphorylated ADP. So that's adenosine diphosphate. So we have the nitrogenous base adenine. So we have an adenine here. Then we have the 5-carbon sugar that's shown here. So this is ribose. So ribose. And sometimes we call this a 3-phosphorylated ribose because we have carbon 1, 2, and 3, and we have this phosphoryl group on carbon 3. And that phosphorylated ADP is linked to the pentothenate unit, which comes from pantothenic acid. So that's this part here. And pantothenic acid is vitamin B5. Next, we have the beta-mercaptoethylamine. So beta-mercaptoethylamine unit here. And then we have the acetyl group on the end. And thus you can see why it's much more preferable for scientists to just write a shorthand like this. So our acetyl coenzyme A. Down here, we see the reaction that converts pyruvate to acetyl coenzyme A. First, pyruvate is decarboxylated, which means that it loses a carboxyl group that is released as carbon dioxide. Next, it is oxidized, and the hydride ion that is removed is accepted by NAD+. Finally, the remaining acetyl group is linked to coenzyme A by a thioester bond. This very complex reaction is carried out by 3 enzymes and 5 coenzymes that are organized together in a single bundle called pyruvate dehydrogenase complex. This organization allows the substrate to be passed from one enzyme to the next as each chemical reaction occurs. This single reaction requires 4 coenzymes made from 4 different vitamins, in addition to the coenzyme lipoamide. These are thiamine pyrophosphate, derived from thiamine, or vitamin B1, FAD, derived from riboflavin, or vitamin B2, NAD+, derived from niacin, and coenzyme A, derived from pantothenic acid. Obviously, a deficiency in any of these vitamins would seriously reduce the amount of acetyl coenzyme A that our cells could produce. This, in turn, would limit the amount of ATP that the body could make and would contribute to vitamin deficiency diseases. Fortunately, a well-balanced diet provides an adequate supply of these and other vitamins. In this figure, we see that acetyl coenzyme A is a central character in cellular metabolism. It is produced by the degradation of glucose, fatty acids, and some amino acids. The major function of acetyl coenzyme A in energy harvesting pathways is to carry the acetyl group to the citric acid cycle, in which it will be used to produce large amounts of ATP. In addition to these catabolic duties, the acetyl group of acetyl coenzyme A can also be used for anabolic or biosynthesis reactions to produce cholesterol and fatty acids. It is through this intermediate acetyl coenzyme A that all energy sources, fats, proteins, and carbohydrates, are interconvertible.